semester because this is more about less of the memorization and biological terminology aspect of things. This is definitely more of an applied aspect. So we're going to start off with population ecology, and then our next section is going to be community ecology. So first, when we're talking about population ecology, we're going to start off with density, dispersion, and demographics. So what is a population? A population is a group of individuals of a single species living in the same general area. So it can be described by their boundaries and their size. So here, this is a map of the United States, and you can kind of see different populations here if we wanted to call these populations. So if we wanted to look at our population of cows, that's in all of the pink. And our population of pigs is around here, it's in the yellow. Lots of sheep and goats there in Texas. This is our sheep and goat population. But you can kind of get the idea. So these are groups of individuals of one species living in the same general area. Density is the number of individuals per unit area or volume. So Saskatchewan has a low density of human population. The province of Ontario has a higher density of human population. So if we're looking at this map again, you can see that there's some, if we're looking at just the pink, we have areas of light pink and areas of very dark pink. The darker it is, then the higher density it is. So there's a lot of cows here and very few cows up here. So different densities. Dispersion is the pattern among spacing of individuals within the boundaries of the population. So what is the pattern of how these different populations are spacing themselves? How are they related? So density, a dynamic perspective. In most cases, it is impractical or impossible to count all of the individuals within a population. So it's very difficult to figure out what the actual density of a population is. So scientists, ecologists have come up with multiple ways to mathematically get estimates on population sizes and population densities. So scientists use a variety of methods to estimate population density, and then we can extrapolate that from a small sample or an index, and then extrapolate that information to a larger scale. A lot of the practice is done is called mark recapture, where you capture a subpopulation of the total population, so a portion of the total, and you mark them. And then you release them back into the wild. Then you go back sometime later and you try and do some more capturing. And odds are you're going to get a proportion of the ones that you marked previously. And you can use that mathematically to determine what the overall population is going to be. What is the ratio of marked to unmarked? So here I have a little description. Scientists capture, tag, and release random sample of individuals, or M for the marked individuals. Marked individuals are given time to mix back into the population. Then we go back and capture a second sample of individuals, or T for total. And now, how many of them are marked, or R for recapture? The population size N is estimated by, so N, our population size, is a equation of the marked individuals times the total captured the second time, divided by the number of recaptures, and that can give you a population size estimate. So here are some different sampling methods. So this is when a mark recapture for birds. So this is why we banned birds, to get unique identifiers, and then we can use this information to do estimates of the population. And then this is kind of a diagram depicting what this description means. So you can kind of piece that out. So we have purple is the total population. Blue over here is first capture. Green is second capture. And then the yellow is going to be the number of marked individuals that were recaptured. So it's just kind of a visualization of this equation. Density is also an interplay between adding individuals to a population and the removal of individuals to a population. So we have things like immigration. Immigration is the influx of new individuals from other areas. So here's our population of birds in the middle, and these birds are immigrating into the population. So immigration here. Emigration is the movement of individuals out of the population. So here we have emigration. They are leaving our total population. And this is also going to be factored in by births. So how many new individuals are being introduced, and deaths. How many are we losing? So immigration, emigration, births, and deaths, all of which are going to be affecting how we measure density. So we have different patterns of dispersion as well. And by dispersion, I'm talking about how organisms arrange themselves in space. So different animals space themselves out in different ways on the landscape. And this can be true for plants as well. So not just animals, but plants as well. Environmental and social factors can influence the spacing of individuals in a population. So we have different definitions for different types of dispersion. We have clumped, uniform, and random. In clumped dispersion, individuals aggregate in different patches. This can be influenced by resource availability and different types of animal behaviors. This is the most common type of dispersion. So here on the right, I have all sorts of different examples of organisms doing this type of dispersion. And if you start going outside and exploring your environment, then you will start to notice these different types of patterns, these different types of dispersion patterns. Next, we have uniform. In uniform dispersion, individuals are equally spaced apart, relatively speaking, equally spaced apart. Generally, this is a result of when interactions between individuals like competition and territoriality are occurring. So emperor penguins up here, they are usually uniform in their dispersion, so they're spacing themselves out evenly across the landscape, and each one has their own little part. We also see this with plants on different landscapes, and then here I have another example of the penguins doing their uniform dispersion things. Next, we have random dispersion. Random dispersion is where individuals are spaced out in unpredictable distances from each other. This is common in wind-dispersed seeds. So here in the top right, we have an example of dandelions just growing every which way that they want to grow. There's no predictable pattern as to how they're growing. And again, this is really common with wind-dispersed seeds. Next, we have demographics. So the definition of demography is the study of the vital statistics of a population and how they change over time. So that is factoring in the birth and death rates. So the tools that scientists use to pick apart the demographics of a population, some of those tools are life tables, survivorship curves, and reproductive rates. So first we're going to go over life tables. Life tables are age-specific summaries of the survival patterns of a population. It focuses on a single cohort or an age group, a group of individuals of the same age at a time. So in our case, like if we're thinking about what demographic or what group of individuals the University of Regina would have, it would probably be the majority of which are people between the ages of 18 to 25. That's usually the cohort group. And then the life tables calculates the percentage of those that survive from one age class to the next. So here's an example of life tables. And I have some ground squirrels here. This has been heavily studied in Belding's ground squirrels. So if we look at this table, we have age, number of alive at the start of the year, proportion alive at the start of the year, number of deaths during the year, death rate, average additional life expectancy in years. So this is just female, so we'll just go ahead and start here. So we have ages 0 to 1, 1 to 2, 2 to 3. And the number alive at the start of the year, we start off with a lot, and gradually they decrease, they decrease, they decrease. And the number of deaths, because this is decreasing, this is also going down. And so you can get a death rate or the survivorship likelihood of our individuals. So there's a 61% chance that the young of the year are not going to survive their first year. So they have a lot of young mortality in this case. And then that mortality rate does gradually decrease. There's a little bit of a spike here. And then there are none that are going to survive past 10 years old. So, yeah. So this is a life table. So what is the likelihood that they are going to survive from one year to the next? Next, we have survivorship curves. Survivorship curves are a graphic way of representing data in a life table. There are 3 main different types of curves in general, type 1, 2, and 3. Many species are intermediates of these, but in general, there are 3 different types of curves. So going back to our ground squirrels, number of survivors on our y-axis and age on our x-axis. So as individuals get older, the number of survivors goes down. And we can see that males are less likely to survive than females, but most of them don't survive their first year, and the likelihood of surviving goes down with age. So here are 3 different types of curves. Human beings are a type 1. We have high survivorship, so many survivors throughout the majority of our life. So like we have just high survivorship throughout our lifetime, and then a steep drop off there at the end. So not, this is going to be like people between 80 to 100, 110 now, because human lifespan is drastically increasing. Then we have our type 2, which is the gradual decrease of a population over time. So it's very linear in nature. And then we have things, our type 3 curves, which have high amounts of mortality throughout early in its lifespan, and barely any that make it past a long or old age. Okay, next we have reproductive rates. Reproductive rates are age-specific summaries of the reproductive rates in a population. They can be used to estimate the number of breeding females, and this does focus on the females within a population. So this is a reproductive table for those same ground squirrels, the Belding's ground squirrels. So we have age in years, so 0 to 1, 1 to 2, 2 to 3. So our oldest individual is 10 years old in this case. Proportion of females weaning a litter. So this is the number of females who have a litter. The young ones don't have litters. And then as we go, we see that 65% have weaning a litter between the ages of 1 and 2. And then we have these guys here that are all weaning litters, pretty high rates. Mean litter size. So this is the number of individuals in a litter. So none for the young ones. And then we can see the peak number of litters are for ground squirrels that are between the ages of 4 and 5. So they're having between 5 and 6 in their litters. It's a little bit lower, just a little for the slightly younger individuals, and a little bit lower for slightly older individuals. So prime reproductive age for the Belding's ground squirrel is between the ages of 4 and 5. Mean number of females in a litter, so this is how many more females are they producing, because the females are going to be having litters of their own. And so again, these reproductive rates and reproductive tables are female focused. We also have different types of population growth. We have exponential growth. This is only going to be under ideal conditions. So everything that these animals or organisms want is at the ready. They have it. They have ideal conditions for growing. So ideal conditions, rate of reproduction is at its greatest. So it's just increasing. They want for nothing. They have everything they need. Exponential growth. However, this is not sustainable. Eventually, all populations will hit a carrying capacity. That is a point where all of the resources are being taken advantage of and the environment or the habitat cannot sustain any more individuals in the population. So eventually, that population is going to hit the carrying capacity, and this is called logistic growth. Growth that is slowed as the carrying capacity, or K, is reached. So here's one that combines these two. This is exponential growth, and this is logistical growth. So hitting that carrying capacity and leveling off. The exponential model describes population growth in an idealized, unlimited environment. Idealized situations help us understand the capacity of species to increase and the conditions that may facilitate its growth. So we have this equation: the change in population size equals the number of births plus the immigrants. So this is kind of going back to that bird picture: births plus immigrants minus the deaths minus the immigrants. For immigration and emigration, or if immigration and emigration is ignored, the population's growth rate is just births minus deaths. This is the zero population growth model. In exponential growth, the population growth under the idealized conditions, the rate of increase is at its maximum, or R max. This results in a J-shaped curve. So this is a J-shaped curve, or a hockey stick-shaped curve. I've heard that also. Characterizes some of rebounding populations, such as the elephants in Kruger National Park, after hunting was banned. So they had very low populations because they were over-hunted up until the late 1930s. And then there were some protective measures put into the park, in Kruger National Park, and all of a sudden the populations of elephants started to skyrocket. And so it resulted in this J-shaped curve. Their population experienced exponential growth because they no longer had the pressures of poaching. The logistic model describes how population grows more slowly as it nears that carrying capacity. An exponential growth is not sustainable. There's only so many resources in the environment, and eventually a carrying capacity has to be hit. A more realistic approach is to incorporate that carrying capacity. The carrying capacity, which is visualized by the letter K, capital K, the maximum population size in the environment, the maximum population size the environment can support, and it varies with the abundance of resources. So depending on the amount of resources in the environment, that carrying capacity could be higher or it could be lower. The logistic model, the per capita rate of increase declines as carrying capacity is reached. So included a new mathematical expression that reduces the per capita increase as the population size reaches K. I realize that I am saying a bunch of mathematical things, and this is a biology class, and I want to emphasize now, I am not going to ask you any type of mathematical question on an exam. I might ask you what K means, I might ask you what N means, but I am not going to ask you to do any math in our exams. So, what does this part of the equation mean? When N is small compared to K, the term is going to be close to 1, and it continues to increase until it approaches the maximum. When N is large compared to K, the term is close to 0, and the per capita rate of increase is low. When N equals K, the population stops growing. So in this case, it's going to keep going up. In this case, it's going to slow down, and then in this case, it's going to be leveled off. So it produces a sigmoidal curve, S-shaped curve. So that is this red line here, our logistic growth model. New individuals are added quickest at the intermediate population size. So intermediate of the population size, it's kind of in this middle area. So population growth rate decreases as n approaches k. So that is this section here. This is where the population growth rate is decreasing because our n, our red line is representing our n for the most part. So population size n, our red line represents that. As we start to get close to that carrying capacity k, this is going to start to decrease. It's going to slow down. It's not as steep of a slope. And then k is eventually reached. Researchers are able to do real-time experiments to look at this type of population growth. We can use single cells or very simple organisms such as paramecium or Daphnia, those small crustaceans, because they can grow in a constant environment without predators or competitors. And so we can actually run experiments to visualize and actually see this in real time. Logistic model isn't perfect, however, but they can be used by biologists to estimate when the critical size below which populations can become extinct. So this was incredibly important to predict when some of our black rhinos were going to be going extinct. So playing into all of this is life history. So life history traits are products of natural selection. An organism's life history comprises the traits that affects its schedule of reproduction and survival. So here I have a little diagram. Life history incorporates all of these. These are all intimately connected. Survival, growth, reproduction. Survival, growth, reproduction. All of these are describing the life history. It's comprised of three main variables. The first one, the age at which reproduction begins. So how early after being born can that organism start reproducing? For humans, that is going to be puberty. When women hit puberty and they start menstruation, then they are becoming reproductive and can start having children. Usually this is around, and obviously I would never promote this ever, but usually around 12 to 13. When you're talking about things like mice can start reproducing within their first year of being alive. So pretty big differences about when different organisms can start reproduction. 2, how often do they reproduce? How often can they produce young? Again, mice are capable of having multiple litters in a year. When you're talking about something like an elephant can only have an offspring every 2 years, I think. They are pregnant for a very, very long time, and then they don't go into a reproductive state again for a number of months, I believe. But I'm not an elephant biologist, but it's a long time before they're capable of reproducing again. And then how many offspring are produced? Mice, again, can have lots of pups inside of a single litter. So many, many pups at a time. Whereas again, an elephant is only going to be having a single offspring. So evolution and evolution and life history diversity. So different environments favor different ways of reproduction. Species that exhibit similarity or Big Bang reproduction reproduces once and then die. So salmon are a really great example of this. They reproduce once in their lives and then die. Species that exhibit heteroparity, or repeated reproduction, reproduce offspring repeatedly. So I put a bear here because bears reproduce every year. Highly variable environments may favor similarity, while reliable, stable environments favor heteroparity. So there's trade-offs. There's trade-offs in life history. Resources are finite, so there's only so many resources in an environment, which may lead to trade-offs between survival and reproduction. K selection, or density-dependent selection, selects for life history traits that are sensitive to population density. So what this is really saying is that how dense a population is affects the amount of offspring that the organism has. Our selection is density independent selection. Selects for life history traits that maximize reproduction. However, and I put this here, this is highly simplified. I have said it time and time again, biology is not black and white, and there are shades of gray for all of these different descriptions that I am giving. So here, this bird example I have up here, this is for a type of falcon. And this falcon can have small broods, very large broods. So let's say 1 egg, 2 eggs, or 3 eggs, where 2 is the quote-unquote normal size, normal brood size. So if they have a small brood size, if they only lay 1 egg, then they only have 1 offspring to take care of, and so the survivorship of both the male and the female is very high, and the survivorship of the offspring is high. For normal brood size, so 2 eggs, you have 2 chicks that need to be taken care of, and so the parents are working a little bit harder, which means they're expending more energy, and so their survivorship is going to go down a little bit. And with a very large brood size, or example 3 eggs, then they have 3 young to take care of, expending lots and lots of energy, and their survivorship is going to go down because they have to expend more energy to make sure that their offspring survive. And the survivorship of the offspring still is not really determined either. So that is an example of case selection. So density dependent, how dense those organisms are is sensitive to the survivorship. So factors that can regulate populations. Many are going to be density dependent, and there are 2 general questions about the regulation of population growth. First off, what environmental factors stop a population from growing indefinitely? What are those significant resources that is going to affect whether or not a population keeps growing or not? Second, why do some populations show radical fluctuations in their size over time while others remain stable? So some populations remain stable and others exhibit this boom and bust cycle. So this is one of the most famous examples out there, the lynx and snowshoe hare relationship. This relationship is intricately connected. So the snowshoe hare is the primary prey source for the lynx. And so when let's start here. Here the snowshoe hare population is very high. And so the lynx population is following it just behind. And it goes into a small spike. But then, because there's more lynx, the lynx eat more hare. And so the population of the hares decreases very rapidly. Now we have a very low hare population. So now there's not as much food for the lynx. So the lynx population also decreases. Okay, well, now we have a small lynx population, which means the hares can survive better. So their population goes, where's my cursor? There it is. Goes right back up again. Now we have a lot of hares, so we have a lot of lynx. So you can see how it's kind of this cyclical boom of a population and then a bust of a population. So population change in population density. In density independent populations, so this is birth rate and death rate do not change with population density. So here is our red line, density independent. So population density is going up, so our population is growing, but it's density independent. So it's just constant. The death rate is still constant. In density dependent, birth rates fall and death rates rise with population density. So this is density dependent. The density of the population depends upon the amount of birth and death rates. Again, I'm not going to throw a diagram like this into an exam. This is to introduce some of these concepts in ecology, just so you can start to get familiar with some of these concepts. I'm not going to make you figure this out. Okay, some mechanisms of density-dependent population regulation. Density-dependent birth and death rates are an example of negative feedback that regulates population. It can be affected by factors such as competition for resources. So if you're competing with other individuals for those resources, then your population density can either increase or decrease. If there's direct competition, your population is probably going to decrease. Territoriality. So if you have territorial individuals, then how dense your population is going to be determined by the size of that territory. Disease. The denser your population is, the more likely disease will spread quickly through a population. This seems particularly potent right now in light of everything. And then predation can also affect how populations are regulated. If you are a prey item, if you're a bunny, and there's lots and lots and lots of bunnies around, well, then you're going to attract some predators. And those predators are going to take advantage of the high densities of bunnies that are around, and it's going to help regulate how many bunnies are out there on the landscape. The more bunnies, the more predators there's probably going to be. Kind of that the lynx and hare thing all over again. And then pollutants and toxic wastes can also regulate population densities. So pollutants can negatively affect these populations and decrease the density. And this is more of an unnatural type of regulation. So looking at some population dynamics, the study of population dynamics focuses on the complex interactions between biotic and abiotic factors that influence the population. There's stability and fluctuation. Long-term population studies challenge the hypothesis that populations of large mammals are stable over time. If we look at, this is an example of moose and wolves. So we have number of wolves here, and so we have two different y-axes. We're looking at two different things. Number of wolves is our blue, number of moose is our red. And this covers over 50 years worth of time. So this is a pretty large time frame. And it's very clear that these populations are not stable over time. As there's lots of wolves, there's low numbers of moose. Less moose means less prey, and so that wolf population kind of crashes. But then, because there's fewer wolves, the moose start to recover, but now there's lots of prey, and so the wolves come back in full and start to decrease that moose population again. So they're very dependent on one another, but they're definitely not stable across time. So again, this goes into the population cycles and how scientists have been looking at this across a number of years. Like I mentioned before, some populations go through these boom and bust cycles. And there's three main hypotheses that help predict this boom and bust. So for this specific lynx-hare example, because it's, again, one of the most studied and most well understood relationships of organisms in population ecology. So the hare population follows food supply is the first hypothesis. That was debunked. That was not the reason why we were seeing this fluctuation in hare populations. Next, we have that hare population is driven by lynx predation. This was supported. So the more hares there were, the better the lynx did, and the more lynx there were. Then, because there was too many lynx, they were eating all of the hares, and so the hare population decreases. You can see it's a cyclical relationship between these two populations. Then there was this bizarre one that that hare populations were driven by sunspots because it changed the environment a little bit, and it was actually supported. I don't remember exactly the data that went into it, but it still kind of seems bizarre to me that hare populations are affected by sunspots. But I think that it was kind of shown that this was just a random association and that they're not really related.